Okay, I think it's a good time to get started. So on behalf of Biocytogen, again, I'd like to welcome everyone to today's webinar. We're very excited to invite experts from our antibody therapeutics research as well as licensing teams to provide insights into Biocytogen's internal R&D programs designed to uncover novel antibodies for future therapeutic development. I'm Jenna Frame from Biocytogen Boston's marketing team, and I'll be moderating today's webinar and Q&A discussion. Joining me on today's panel is Dr. Vivian Tian, who is the Senior Director of Business Development and Licensing and serves as the head of the Biocytogen Europe Innovation Center in Heidelberg. Dr. Tian has led numerous antibody and run mice licensing deals for Biocytogen over the past few years with companies including Merck, Janssen, ADC, Therapeutics, and multiple others. And finally, I'd like to introduce today's speaker, Dr. Frank Ahn. As the Senior Director of Antibody Therapeutics, Dr. Ahn's key responsibilities include overseeing Biocytogen's internal antibody discovery programs, including portfolio strategies, target selection, and project implementation and management. With 2 decades of experience, Dr. Ahn has strong expertise in pharmacology, pharmacodynamics, and cell-based and biochemical assays used to advance multiple drug modalities, including small molecules, peptides, and antibodies. So without further delay, I'd like to invite Dr. Ahn to begin the presentation.
Thank you, Jenna. You can hear me all right, right?
Yep, we can hear you great.
Okay, great. Thanks, Jenna, for the nice introduction, and welcome to everyone here who came to the webinar. Today, we're going to talk about the bispecific antibody-drug conjugates, or in short, bispecific ADCs. Okay, so I'm going to talk in 3 parts today. First, I would like to give a brief introduction of our company, Biocytogen, and its antibody discovery platforms, as it is the foundation of the data, the bispecific ADC data that I'm going to tell you about. And I will also give a brief review of where the bispecific ADC field is by introducing you to some of the very recent clinical data that were reported in recent scientific conferences. Then we're going to spend the majority of our time talking about Biocytogen's bispecific ADC programs. I'm going to show you 2 very nice data sets that indicate some of the activities of our bispecific ADC programs, molecules. First, about a brief introduction about our company, Biocytogen. Many of you might have already heard or known Biocytogen if you used our humanized mice or other mouse strains or our pharmacology CRO services. In a nutshell, Biocytogen is a public clinical stage global biotech company with over 1,400 employees, headquartered in Beijing with offices in Shanghai and the Jiangsu provinces. And also we have a site in Boston, US, and an office in Heidelberg, Germany. So we have a good clinical pipeline. We have 6 clinical assets that you're going to see on the next slide. And we are very active on the preclinical stage through an effort we call the Project Integrum, about early antibody discovery efforts, and we have so far over 600 programs that are either completed or are ongoing, and more are being worked on as well. We're also very active in collaborations and the partnerships, licensing opportunities. So as of the end of June, we have 50 molecular licensing agreements and 42 platform licensing agreements in place. This shows you our clinical pipeline. We have 6 molecules in clinical trials, 2 in phase 2 and 4 in phase 1. Among them, is one ADC asset that, as a collaboration with RemeGen, is a Claudin 18.2 ADC. And we also, just very recently, this year in February, we put in collaboration with Weixin, and we put this bispecific molecule into clinic. At BsADC antibody discovery, we have been working on these different modalities of antibodies. We worked on very extensively on monoclonal antibodies on our RenMab mouse platform. We have a bispecific antibody platform based on our RenLight mouse strain. This is a common light chain bispecific antibody platform. And we also have a nanobody or single domain nanobody platform we call based on our RenNano mouse. You're going to see an introduction about these mice in the next few slides. And we have a bispecific ADC platform. We worked on TCR mimics and also GPCRs, targeting GPCRs. All these different modalities are based on the RenMab, RenLight, and the RenNano mice. Collectively, we call them Ren mice here, indicated here. Ren in Chinese means human. So these are fully human antibodies. Okay, so because all of our antibodies are from this platform, these mouse strains, so I'd like to introduce them to you briefly in the following few slides. So this slide shows how the RenMab mouse was constructed. So on the immunoglobulin heavy chain side, we replaced the mouse heavy chain locus, the V region, with the entire human heavy chain locus in situ in one replacement. So in this, in the heavy chain locus, we have the entire human heavy chain repertoire here. That's the heavy chain. On the light chain side, we use the entire human kappa chain locus and replaced the mouse kappa light chain locus. So in this, this mouse also carries in the entire human kappa chain repertoire. So altogether, this RenMab mouse strain carries the most complete human heavy chain locus repertoire and the human kappa light chain repertoire. We use these RenMab mice extensively for our antibody drug discovery. And one of the things we use these the RenMab strains on is an effort called a Project Integrin. It is an early antibody discovery effort, and we're trying to discover antibodies that are against over 1,000 potential targets. We're motivated doing this because there's a gap between how many targets have been used to generate drugs in the clinics and how many potential drug targets there are. That includes the surface molecules and also secreted molecules. So there's a huge gap, and we would like to fill this gap by discovering a lot of the antibodies against a lot of the targets. This is a snapshot of where we are with the Project Integrin. This is ongoing effort. It just shows that currently we have over 150 immunizations going and over a couple of hundred antibody screens going with over 600 hits. These are program hits. And also, as I mentioned, 50 partnerships out of this early antibody discovery effort. This is give you a flavor of some of the targets we have been working on. These are organized by disease areas, oncology, immunology, etc. This is not a complete list, and if you are interested in some of the targets of your favorite targets, and please feel free to contact our BD team, and they can provide you with a more complete list. So then the second mouse strain we use for our antibody discovery is the Ren-like mouse. This is a common light chain platform that is ideal for bispecific antibody discovery. So this slide shows how it was constructed. On the left side is the mouse, is the heavy chain locus, antibody heavy chain locus that was constructed identically to the Ren-like mouse. However, on the light chain side, it's different. We use just one fixed kappa light chain gene, 3-11, and to replace the entire mouse kappa chain locus. So in this mouse, all the heavy chains are forced to pair with this single fixed sequence light chain, so that all the light chains are common to all these to all the functional antibodies. These Ren-like mice, although they only have one common light chain, but they can generate as robust immune responses as the wild-type mice. And they can produce quite a diverse and high affinity antibodies, and they carry very few somatic mutations on the common light chain. So these properties making them a very ideal format for bispecific antibodies, because it's addressed a very challenging and historical and also frustrating issue when producing these bispecific antibodies, which is the mispair of the heavy chains and the light chains. If you, as illustrated in this diagram, if you have a common light chain, as indicated by these gray bars, if you have a common light chain, in the expression system, you only need to express two different heavy chains and this common light chain, and you get vast majority of the product is the desired functional bispecific antibodies. And this is in contrast to if you have two different heavy chains and two different light chains, and you express them in a single cell, and then only a fraction of the antibodies would be the desired functional bispecific antibodies. And this is, of course, you have to use the heavy chains also needs to be assembled correctly, and our common light chain antibody platform is compatible with a number of heavy chain assembly technologies commonly used in the industry. One key feature in Biocytogen when we engaged in antibody discovery is to use the knockout mice in both the Ren-mab and the Ren-light background. This is mostly is that we want to break the immune tolerance of the very highly conserved epitopes. So as we all know that there's a certain homology or a range of homology between mouse and human proteins. As illustrated here, the green colors indicates the highly conserved regions between human and the mouse. So if you have a very highly conserved protein, a human protein, relative to the mouse, then this protein would be less immunogenic. So in order to break this immune tolerance, there are people who have taken different approaches. Some people would go to more remotely related species like chicken But at BioCytogen, our approach is to just knock them out in the mice so that the mouse would not see the human proteins as known molecules. So that would help expand the pool of epitopes. As illustrated here, we performed identical immunizations of a target in both the Ren-like wild-type mice and also in the Ren-like knockout mice where the target was eliminated. So as you can see, we got more clones after beacon selection. That indicates that there's more immune responses with the knockout in the knockout strain. And also when we look at the germline gene usages, and we can see that with the knockout, there's certainly more diverse germline gene usages. These are heavy chain germline gene usages. So we routinely employ knockouts in our antibody discovery as long as the targets we are trying to knock out are not lethal in these mice. And finally, this is introduction. This is about Ren-nano mouse, how we constructed that. This is the most recent addition to the Ren mice family. On the heavy chain side, again, we replaced the mouse heavy chain locus with the human, entire human heavy chain repertoire, the V region. The difference is that we removed the CH1 of the constant region from the mouse genome. So in this mouse, the heavy chain side, there's the V region and CH2 and CH3, but CH1 is missing, so that this molecule would not be able to pair with a light chain molecule. So this the final result is a single domain nanobody. We call that nanobody because the size of it is smaller than the traditional antibodies. And we have started a initiative called the Nano 100, where we are attempting to generate nanobodies against over 100 high-value targets. Here, high value means these targets are either have active drugs against them, so they are they have clinical values, or there's active clinical programs targeting these targets. So we're trying to generate these single domain antibodies against over 100 of such targets. Okay, so that was the introduction of our antibody discovery platforms. Now we're going to turn our attention to bispecific ADCs. So what is bispecific ADCs? So it's very simple. It's just that bispecific antibodies plus a linker and a payload. So as illustrated in this diagram, the difference between bispecific ADC and classical monoclonal ADC is just the number of targets this one single molecule recognizes. So in bispecific ADCs can target 2 TAAs, tumor-associated antigens at the same time. And at the Biocytogen, the difference we think the advantage of using the Biocytogen common light chain platform is that this bispecific targeting is mediated by a common light chain bispecific antibody. And the one major advance of using the common light chain is at the CMC stage. It's very convenient to express a bispecific antibody with high yield. So why we like bispecific ADCs? Because these molecules provide certain advantages over the classical monoclonal ADCs. They can improve efficacy in terms of improving the internalization efficiency and cytotoxicity against the tumor cells if you choose the two arms carefully and correctly. And it also helps reduce resistance. This is mainly because you are hitting the tumor cells with two targets and at the same time instead of just one. And we also believe it should also help tumor targeting and then reduce the off-tumor on-target side effect that was the issue of some monoclonal ADCs. Although there are increased interest in the bispecific ADCs, it is still a field that is just taking shape and in its early stages. So there are not a huge number of bispecific ADCs in clinic, and there is just a few. And one of them is the by AstraZeneca AZD9592 that targets EGFR and the c-MET. And they just introduced this into clinic at the end of last year and probably started dosing first patients earlier this year. And the other molecule that was just reported in this year's ASCO last month is this molecule from Bailey that targets EGFR and HER3. They really, reported some exciting data, and this is the molecule that is a bispecific. It's a 2x2 format, anti-EGFR and HER2, with a exatecan-like payload and a DAR of 8. They optimized the targeting arms against EGFR with a high affinity, and the anti-HER3 and with a low affinity. And the overall, they tested this on non-small cell lung cancer and a few other cancers, and the overall response rate was very impressive at the 45.3 ORR. So to put this data in perspective, if we compare this bispecific targeting molecule, EGFR and HER3 versus this HER3 targeting monoclonal ADC, and the authors split the response in small cell lung cancer into two patient groups, EGFR wild type and EGFR mutant. And you can see if you compare in both categories, in the EGFR wild type situation, the ORR in both situations, the ORR for this bispecific ADC was more favorable compared to the HER3 single targeting ADCs, almost 45 versus 28, and 63 versus 40. So of course, this is a cross-trial comparisons, and you should take the comparisons and these results with a grain of salt. But there's encouraging indications that in this, at least in this particular scenario, the bispecific ADCs appears to be more active. So this is actually the first clinical evidence that bispecific ADCs shows better anti-tumor efficacy and with equivalent adverse events profiles. This is just very quickly to show that there was the increased interest in bispecific ADCs. There are more molecules that are going into clinics. Here, this table just shows a few molecules that are currently in clinical trial stages. That includes, two different targets and also the biparatopic ADCs as well. This is just to quickly say that bispecific ADCs has been emerging interest in recent scientific conferences, and also in the ADC field, people are also interested in novel format of targets that payloads in terms of dual payloads, immune activating payload and radioactive payloads. And also recently that RNA delivery, targeted RNA delivery by antibodies has also gained momentum, particularly generated a lot of interest by investors. And I just wanted to point out that the antibodies used for the RNA delivery can be monospecific or bispecific antibodies as well. So that was just a quick overview of the bispecific ADC field. And now we're turning our attention to two specific cases that we have been working on at Biocytogen. So the first bispecific ADC case study is a molecule we codenamed YH12 that targets HER2 and TROP2. This shows the structure of this molecule. On the left is the targeting moiety that targets HER2 and TROP2 by a common light chain antibody. And we used the classical vc-MMAE as the linker and the payload with a DAR of 4 for this molecule. For our studies, we, in addition to generate the final molecule, the bispecific ADC molecule, we also generated the antibodies or the ADCs for the parental monoclonal arms that against HER2 and the TROP2, and also the monovalent parental antibodies, the single arms against HER2 or TROP2, because we would like to compare all these molecules in our studies and demonstrate the improvement by in terms of activities by our bispecific ADC targeting. Just a quick word about the rationale for us to take to find this TROP2 and HER2 combination. This is because both molecules are highly expressed in certain breast cancer populations, and also, as we all know, that in the recent trial, the anti-HER2 monoclonal ADC also demonstrated great OS benefit, even in the HER2-low, the majority of the breast cancer patients population. However, if you look at the trial data from Destiny Breast 04, the ORR for the monoclonal single targeting ADC is just a little above 50%. So that's indication that there's also half of the patient populations did not respond. So our thinking was that by targeting both HER2 and the TROP2, by the way, both targets have antibodies, monoclonal ADCs approved targeting these two molecules, these two targets. And our hope is that by putting them together on a single molecule, we can improve this bar, push this bar further higher so that more patients could benefit from the therapeutic treatment. So I'm going to tell you a series of data about this molecule with a common theme that this molecule performs better than the monoclonal ADCs or the monovalent parental ADCs. So let's look at the binding. So let's first look at the panel on the top left. And here we have two cell lines, NCIN87, which is a double positive for HER2 and TROP2 cell line. And also we have another cell line, MB468, that is a single positive only for TROP2. So if you stain a mixture of these two cell lines with a regular TROP2 monoclonal antibody, then you'll see a rightward shift almost to the two similar positions in these two cell lines. However, if you stain the same mixture of these two cells with a bispecific antibody, and you see a much rightward shift with the double positive cells versus the single positive cells. So if you do a ratio of the signals of double versus single, and you plotted the data, you get this panel here, and the purple bar indicates that the YH12 binding, okay, more favorably to the double positive cells. And if we do the opposite side of things, is that if you look at this cell line SNU5, that is a single positive cell line for HER2, and you plotted the data, and you can see when the bispecific molecule YH12 binds more preferably to the double positive cell line N87 versus SNU5. So this is binding. This slide shows you the internalization data. This here is the live monitoring of the internalization signal over 24 hours in InQSite. So the assay we used is we labeled a secondary antibody that would be internalized together with the primary targeting antibody into the lysosomes and introduce a pH-dependent color change and emit fluorescence. So we could follow the fluorescence over time. So this red line is the naked YH12 molecule. And it's indicate it has a much more efficient internalization activity versus here is the two blue lines are the parental anti-HER2 and anti-TROP2 monoclonal antibodies, all the parental monovalent HER2 and TROP2 antibodies, the two green lines here. And if you tabulated the data at a given time point, for example, if you just look at 24 hours, and this high red bar is the YH12 internalization signal, and these two green bars are the monovalent or single arm parental HER2 and TROP2 internalization signals. So clearly you can see there's a certain level of synergy with the bispecific molecules versus the single arm molecules. This is the in vitro sex toxicity data of YH12, which is the red line here, and it's more potent in terms of inhibiting the N87 cell growth versus the two parental monovalent ADCs against HER2 or TROP2 separately. This is in vivo CDX data against this cell line, NCIH 1975, which is a double positive for HER2 and the TROP2 at intermediate level of expression of these two TAAs. So YH12 is the orange line that is most potent among all the molecules we tested in this study, and more potent relative to the monovalent parental anti-HER2 and anti-TROP2 ADCs, the two green lines here, and the two pink purplish lines, which indicates the monovalent anti-HER2 and anti-TROP2 ADCs here. And this is, again, the NCIH 1975 xenograft model. But this is a different twist from the previous in vivo study. So here, as you can see, first is that YH12 showed a dose-dependent inhibition of 1975 tumor cell growth. Okay, and then if you look at the YH12 at the 1 mg per kg dose, which is this darker orange line, and if you inject the mice, okay, with the same amount of paratols, but split the paratols to 2 separate molecules and co-inject these 2 separate molecules, ADCs, into the mice, as indicated by this lighter blue line, you can see that the bispecific molecule is more potent than a combination of 2 separate molecule, the 2 separate ADC molecules, even though there are the same moles of paratops in the mice. So that's this data clearly indicate there's something synergistic about a bispecific targeting versus even a combination of a single TAA targeting. And so here this slide shows the PDX, the efficacy against the PDX model. It's the pancreatic PDX model. And then again, YH12 is the most potent in this model, and in this model relative to the 2 parental monoclonal ADCs, the green lines all. Yes, they are okay. The parental monoclonal ADCs, and also we also tested this anti-TROP2 and 2 anti-HER2 clinically approved drugs. These are actual drugs in the same study, and our molecule was more potent than those approved drugs that are targeting targeting TROP2 and HER2 separately with some monoclonal ADCs here. Of course, this model is in the HER2 high and the HER2 and the TROP2 high PDX, and we are mostly interested in this molecule if it can work in the HER2 low model, as this in the breast cancer is the largest breast cancer patient population. So as you can see here, okay. As you can see here, that if we compare the same dose level, here is the lighter orange line is the YH12 at 3 mg per kg, and that compares favorably to the 2 parental monoclonal anti-HER2 and anti-TROP2 ADCs, or the monovalent parental anti-HER2 and anti-TROP2 ADCs. So that indicates even in the HER2 low situation, our molecules also performed really well. And this is again, when in this particular model, when you stack them up, our molecules also performed better than the approved anti-TROP2 and anti-HER2 drugs. ADC drugs. This is a crude safety and also stability test of YH12 in mice. Here is that we use the C57B6 mice and injected YH12 all the way up to 90 mg per kg. And you can see in this mouse strain, there's no change of body weight, even up to 90 mg per kg. And this is in contrast to if you inject the mice with the equivalent molar equivalent, same molar equivalent payload, only the free payload, and you can see a quick drop of body weight in the first few days. So that indicates that first is the stability of these molecules are quite stable in this mouse strain, and also that this molecule is probably not causing a huge toxicity to these mice. Of course, in these mice, the target, the human TROP2 and the human HER2 were not present. So we also tested the same doses in the humanized mouse strain that co-express both HER2 and the TROP2. And you can see here that at the highest dose, 90 mg per kg, you see a gradual body weight decrease. However, at 30, the body weight remained constant over the first week after the injection. So that's an indication that this molecule, even in the presence of its targets, was quite stable and reasonably lack of toxicity to this strain. And here is shows some developability assays we have done on this molecule. Here is the stability data where we tested this molecule under different stress conditions, like raised temperature for several days, frequent freeze and thaws, and a change of pH to a much lower pH point, and monitored the integrity of YH12 with several analytical methods. And as you can see here, that in all the conditions, stress conditions, and by all the analytical methods, the molecules remains intact. So that indicates a good stability of these molecules. And recall, this is a Bispecific molecule generated from our common light chain antibody discovery platform. Also, just to show you that we were not just being lucky with YH12, and I'm going to show you very quickly about another Bispecific ADC molecule, YH13, that target EGFR and c-MET. And pretty much it is a very similar story to that of YH12. So this YH13 targets both EGFR, targeting the c-MET and EGFR. And again, this is by our common light chain antibody format, and it's a VC-MMAE linker payload with a DAR of 4. And the pairing of targeting of both MET and EGFR makes good sense, because not only because that both targets are highly expressed, widely expressed in a number of tumors, but also MET is is one of the known resistance mechanisms towards EGFR inhibitors. This data shows in terms of binding to a double positive cell line on 1975, YH12, the orange lines, as either the naked molecule or the actual ADC, binds this double positive cell line preferentially versus the parental monoclonal antibodies or the parental monovalent single-arm antibodies. That's the green lines and the purplish lines. This is the internalization data. YH13 is the orange line on the top, and much more efficient in terms of internalization versus the two green lines that indicates the parental monoclonal antibodies, or the two purplish lines that indicate the monovalent anti-EGFR or anti-c-MET antibodies. This is similar to the Pareto split study that I described to you with YH12, and here you have YH13 at 1.5 mg per kg, and if you split the Paretos into 0.75 mg per kg parental anti-EGFR monoclonal ADC or the parental anti-MET monoclonal ADC, the Bispecific molecule won out over the combination of the monoclonal ADCs, which is indicated by the blue line here. This is, again, two different PDX models where the YH13 molecules show the potent inhibition in a dose-dependent manner. These are the, again, the developability assays, purity, stability assays at the different temperatures, different pH, freeze and thaws, and you can see the integrity of this molecule was intact. So that indicates that our common light chain bispecific ADCs are very good for the actual CMC development. At BsADC, we have been putting a lot of emphasis on bispecific ADCs, and we have been working on over 200 TLE targets. Here's a small sample of them here, and on the right and on the left, here is some of the combinations we constructed and the various testing stages of bispecific ADC molecules. So all these are based on our common light chain antibody platform. And for a lot of these targets, we already have clones. So it's a matter of fact, to design to know your biology and select the targets that targets your biology, and the rest is pretty much a very straightforward plug and play. So you can construct these bispecific ADC molecules and test in mice for efficacy studies. Okay, so I would like to conclude my presentation with this slide that just indicates that we believe we are very strong in antibody discovery and making bispecific antibodies, ADCs, etc. But we are also very open to collaborations in the ADC field with companies that have expertise in other areas such as linkers and payload, so that could better dovetail with our antibody discovery strengths. Also, we're very open to out-licensing opportunities, either for our bispecific ADC molecules or for, like I described to you, our early antibody discovery efforts, Project Integrin, and so we're very interested in collaborations and out-licensing opportunities there as well. And if you have any questions, please feel free in the Q&A session to our senior licensing director, Vivian. So with that, before I yield the mic back to the moderator, I just wanted to thank the project teams that generated the data that I described to you today, and thank you very much for your kind attention to my talk. Yeah, so if time permits, I'll be happy to take any questions. Okay. Thanks, Jenna.
Thank you, Frank, so much for that fantastic presentation. I think it was very informative. So I'm going to turn to our Q&A session and start to address some of these questions, and I'm sure we'll have enough time to address most of them. Okay, so the first question is looks to be related to the run nano. So when the constant region genes are replaced by a single one in run nano, are B cell maturation steps affected or somatic hypermutation affected?
Okay, I got the second part of the hypermutation, and so the REN-like common light chain, as I mentioned, carries few somatic mutations on the common light chain. We do find sometimes mutations here and there on the common light chain, but more often than not, these are bona fide common light chains. In other words, there's no somatic mutations on these common on the light chains. Sorry, I didn't get the first part of the question. There was another point about these.
The question was about B cell maturation in the Ren nano model.
Oh, cell maturation. You mean the antibody maturation, if I understand that correctly? No, there's no difference. We have done extensive characterizations of these mouse strains. In terms of the B cell development, they are quite normal, just like the wild type, the B cells in wild type mice.
Okay. All right. Thank you. Okay, so the next question, do we need the bispecific antibody, does it need to bind both targets to be effective?
Okay, it's not, strictly speaking, the bispecific antibodies does not have to bind two targets at the same time in order for it to work. As I showed you that the monovalent ADCs also worked. It worked much weaker. Okay, so less well than the bispecific ADCs.
Okay. So the next question is related to, let's see, the mouse CH region. So do you remove the mouse CH region at some stage to generate a fully human antibody or a bispecific antibody, or is that the sponsor responsibility?
The H region?
I assume it means I assume what we're talking about is the constant region to generate the fully human antibody. So swapping out the mouse constant region with the human.
Yes, okay. So the constant region in the Ren mice, these are mouse constant regions. This is mainly for the developmental considerations. In other words, we wanted the B cells not to generate that these clones will develop well inside the mice. However, as soon as we screen them and we know the sequence, we can make different FC regions and together with the variable regions to form the fully human antibodies, antibody molecules, and we have choices of introduce any mutations to the FC engineering if you so desire.
Okay, thank you. Okay, so I think we have a couple of specific questions about the introductory material. One of them mentions for HER3 single treatment, EGFR mutation was used as a criteria to divide the groups. I believe this was about the clinical data that you presented at the beginning. I don't know if we need to.
What was the question?
For HER3 single treatment, EGFR mutation was used as a criteria to divide groups.
Yeah.
Can you comment on that, or?
Oh, okay. Yeah, so as we all know that in small cell lung cancer, EGFR mutations is one of the key drivers for this type of cancer. So traditionally, people with EGFR activating mutations are treated with the small molecule drugs. And there's very effective small molecule drugs targeting EGFR. However, these patients would eventually, relapse, and but that is a distinct patient population. So that's the mutation I referred to, and the opposite side of that is the wild type EGFR population. So I use the word wild type here is actually precisely is that these are the non-activating EGFR molecules. So that's a different patient group. So in clinical trials, they separate these two things because there's a different clinical strategies for these two populations of patients.
Okay, I see. Thank you. Okay, I think there was one question, again, addressing about whether combinations of the combination of the two different antibody treatment is as effective as monoclonals. Can you comment on that? I know you showed some data arguing that the combination is more effective.
Okay, so let me try to use a different word, combination, because sometimes it's confused with combo therapies where you put the two molecules at the same time, either in the mice or in patients. So here is that instead of this, you put the two separate molecules for bispecific ADCs or molecules, they are actually a single molecule, but targeting two different targets simultaneously. So these are one molecule, the two arms are always go together, versus if the two targeting moieties are on two different molecules, there might be different distributions of these two separate entities. So that's the difference. So in our data, in our hands, we have shown in both cases that the bispecific molecules work much more efficiently than the two separate molecules present inside the body at the same time.
Thank you for clarifying. I think that's a great answer. Okay, so next question. Have you looked at different ranges of expression of the TAAs, HER2, and TROP2 in your PDXs, such as TROP2 low, HER2 medium, TROP2 medium, etc., HER2 low? Is YH12 more efficacious in low, medium, or high expressors, or do you see differences based on the levels of TAA expression?
Okay. Yeah, so we tried to test in all the different scenarios of combinations of high, low, lows, but we were limited by the availability of the models, like particularly in the PDX situation. So we tested something that we can get our hands on, but by no means it is exhaustive of different scenarios. But I think this is still a work in progress, and if we later can get one of those different combinations in terms of expression levels, and we would be glad to test our molecules in those models as well.
Okay, that sounds good. Thank you. Okay, so next question. I think we'll try to get to everything. I know there's a few more that have come in. Hopefully, we won't go too far over. So thank you for staying with us. So could you please comment the ADC conjugation outcome in terms of the procedure and difficulties versus monospecific ADC?
Oh, in our hands, basically there's no significant or no difference at all between conjugation part for a bispecific versus a monospecific ADC, particularly with our Enlight platform. These are just natural antibodies, and except they just happen to have two same light chains.
Okay, great news. So next question. What challenges do you see in using the preclinical data to predict human responses for bispecific ADCs?
Oh, okay. That's a truly $1 million question, or even could be $1 billion question. We do our best to test our molecules in different PDX and CDX models, but these are still models. Whether they can translate directly to human in terms of efficacy, it is a question mark. It depends on the different targets, different tissues, different indications. Truly, I don't think there's a unified or straight answer to that. Another point is safety. Clearly, we would not be able to address safety in a very translatable way in mice, and that's something that we're still working on. So yeah, I mean, to be sure that we know the mouse models will help, but they won't answer all of our questions.
Yep, definitely a challenge in the field. Okay, so next question, how do you screen the bispecific antibody backbone? Any selection criteria for affinity or internalization?
Our main selection criteria for bispecific ADCs is internalization. This is a similar strategy employed by other companies as well. But at the Biocytogen, we do a large scale screen. We can screen, let's say, even hundreds of combinations very quickly in terms of our internalization assays. So we choose the internalization, the better ones, and also we are trying to also address the safety concerns, the off-targeting, off-tumor targeting side of things. So we also select certain combinations that would help us address concerns in that area as well.
Okay. Next question. Have you compared the efficacy of bispecific ADC with parental ADCs in single TAA positive models?
That's a very good question. We might have done that in some other programs, but honestly, we don't do that very often because we have done something equivalent. In other words, we have a monovalent ADCs or single targeting ADCs on the double positive cells. So it is a kind of equivalent situation where those ADCs can only bind a single target on the cells. And our data indicates that in that situation, the monovalent ADCs would not work as well as the bispecific molecules. We did not do exactly like the audience question was about, because when you're talking about the two different cell lines, and when you need to compare the efficacies, it's tricky. The studies needs to be very well controlled in order for you to make the comparisons. So that's one of the reasons we didn't do that very often, but we routinely test our bispecific ADCs and monovalent ADCs against the same double positive cell lines.
Okay, thank you. Okay, next question. In Ren mice, it appears that CH is retained. How much of a concern is this for ADA? I would say in terms of ADA, first of all, ADA happens without a really kind of unknown reasons, and sometimes you see that happen. And I would say this is that it happens to our molecules and also to a lot of our control molecules that we use in our studies. So it's not a very frequent phenomenon, ADA, but we did observe them, but it didn't strike us that our molecules was any different than some of the control molecules we used in terms of the ADAs, even though a lot of the control molecules we used are in clinic, so they're supposed to be optimized to reduce the appearance of ADAs. Okay, thank you. Right. A few more questions to go. So it was mentioned that bispecific ADCs can reduce off-tumor on-target toxicity. However, for biparatopic ADCs, this should not be the case, right?
Yes, because I think that is true. Because you can argue that on the tumor there's usually the presence of the two TAs, but in normal cells, some normal cells only express a single TAA, so the targeting would not be as well. But as long as the single TAA is present, then the biparatopics would target. Yeah, so I think that was the right inference, but I'm just not aware if there was any data to directly support that argument. But in theory, I agree with that point.
Okay, thank you. Is there any advantage conjugating a payload to CH1 rather than CH2 or CH3?
Okay. The way we conjugate is use cysteines. So there's the cysteines like interchain and between the heavy chains and between the heavy chain and light chains. So these are the disulfide bond that could be easily disrupted and exposed to allow conjugation. So mostly, when people use cysteine to conjugate it, that's where the payload would go. So they would not go to CH3, for example. But in order to target CH3, you have to use other ways to introduce a conjugatable site. So that's not something that we're doing, but there are a lot of people working in very creative ways, trying to attach the payload to different parts of the antibodies. And there have been reports that the different conjugation sites would have different properties for these molecules in vivo. So it's just we're not doing that, but there are ways to do that out there.
Okay, thank you. I think we kind of covered how we select the TAA target, so I'm going to skip over that one for now due to time. Let's see, how does the developability of the bispecific antibodies compare to its parental clones?
Oh, okay. We know the yields can be similar. In other words, our bispecific molecules are very friendly to the CMC processes. Of course, I mean, I cannot say for every single molecules, but just say those we selected for further development into the CMC stage, those we can generate yields as good as several grams per liter. So if that's in the acceptable CMC development criteria.
I see. Thank you. So since there was another question about the linker and payload, have we, besides the VC-MMAE, have we tried any other linkers or payloads?
Yes, we know that VC-MMAE is a classical linker payload combination and has appeared on several approved ADC molecules, but it has its drawbacks and the limitations and toxicity profiles. So we're trying to improve upon those blemishes of this linker payload combination. I'm not sure that I'm allowed to say anything in specifics, but just say we are working on different linker and payload strategies for our lead bispecific molecules.
Okay, good to know. Thank you. Let's see. So there's a couple of questions about clone selection criteria and target selection. I think we've covered a lot of that, but I don't know if you want to add any final comments about clone selection criteria, how we're picking clones to construct the bispecific for ADC development, or selecting the tumor targets before we sign off.
Well, in terms of target selection, there's a few things that you do need to pay attention to. I think the first is biology, because you wanted the two targets are relevant to your disease, and ideally they would be validated targets if possible. And then they need to be co-expressed at the reasonable levels in tumors, and also you should avoid those that are also co-expressed in normal tissues. So these are some of the main considerations in target selection. I think I've talked, several points about the screening. You do need to have a large, high throughput, reasonably high throughput screens to select those two arms. And so that's something that just to bear in mind, that not all the clones you can put the one together that would equal two in this situation. So you do need to select the two arms.
Okay, thank you. Let's see. Any further comment on balancing the affinities for the two TAAs?
I think at the BsADC we do pay attention to affinities, but we'll also pay more attention to internalization efficiencies, because that's sounded to us is a more direct metrics for measuring the activity of those ADC molecules. But we do pay attention to the binding properties of the two arms.
Okay. Okay, so I think we got time for just one or two more. So how are we addressing the challenges associated with manufacturing and scalability for the bispecific ADCs? Any strategies or technologies we're implementing to ensure efficient production and commercial viability?
I guess I kind of touched upon that with the previous question, or one of the previous questions, and that is that our common light chain platform is quite CMC process friendly. So we do the normal things that are necessary to optimize the yield, etc. But we have not seen any particulars with those common light chain platforms. In other words, they perform quite similarly to just a regular routine monoclonal antibodies in terms of our CMC process.
Okay, thank you. Okay, I think some of the other questions touch upon topics that we've already discussed, but I would definitely encourage attendees, if they have further questions, to please reach out to us either via our website, and you can contact our licensing team at bd-licensing@biocytogen.com. And we will reach out to all of the attendees after the webinar concludes later today with a link to revisit the webinar if you'd like to view the content again. So I think in closing, I'd just like to again thank our speaker, thank our panelists for joining us today, and wishing you all a great rest of the day.